Adequate nutrition is crucial for amyotrophic lateral sclerosis patients. As the disease progresses, the patients gradually lose the ability to chew or to swallow with ease. At the same time, the abdominal and pelvic muscles weaken, often resulting in depression. Patients often lose the ability and the desire to eat, making malnutrition a common problem. The recognition that aggressive nutrition intervention is paramount among the ALS patients has spurred ardent research efforts aimed at elucidating the potential therapeutic value of dietary supplementation. Ultra-high 25 mg per day for a month intramuscular dose of methylcobalamin, a form of B12, has been shown to slow muscle wasting. Low levels of B12 have been associated with nerve damage in many different animal models. One of the main problems associated with low levels of B12 is elevated levels of methylmalonic acid, and this is toxic to neurons. Low levels of B12 are also associated with poorly functioning peripheral nerves, and this can be exacerbated by ALS. Vitamin B12 can also prevent the damage to the ophthalmic nerve by reducing the MMA and the homocysteine levels, both being associated with oxidative damage. Low levels of B12 have also been associated with neuronal degeneration in other models. Mutations to copper and zinc oxide dismutase gene are responsible for 2 to 3% of ALS cases. These mutations result in the SOD enzyme having a reduced affinity for zinc. In fact, the loss of zinc from SOD1 results in the remaining copper in the SOD1 becoming extremely toxic to the motor neurons. Altering zinc levels within the brain is being studied as a method for treating many different nervous system diseases, including ALS. However, a study conducted at the Linus Pauling Institute found a large dose of zinc inhibited copper absorption, and this can lead to anemia. In the study, researchers added a small dose of copper to animal ALS models receiving zinc, and they found the copper prevented the early death associated with high doses of zinc. In summary, adding a small amount of copper to the subject's diet prevented this lethal anemia, suggesting moderate amounts of zinc supplementation combined with small amounts of copper might help to prevent neuron death in ALS. In animal models of ALS, ginseng was shown to significantly delay the onset of the ALS symptoms. An extract of the ginseng plant, called ginsenoside, has been found to increase the expression of SOD1. Ginseng and its extract might also be able to protect the motor neurons from apoptosis and membrane damage, further helping to slow the progression of ALS. Gingko biloba has antioxidant properties. Additionally, it's been shown to promote healthy mitochondrial function. During an in vitro study, it was found to protect against glutamate-induced excitotoxicity. Gingko also reduced the weight loss in a mouse model of ALS. Gingko extract has been shown to protect neurons from death due to oxidative stress. CoQ10 acts as an antioxidant and is essential for the proper mitochondrial functioning. Human studies have found ALS patients have a higher percentage of oxidized CoQ10, a condition the researchers blamed on oxidative stress caused by the disease. Supplementation with ubiquinol, the reduced non-oxidized form of CoQ10, might help to ameliorate this problem. In an animal model of familial ALS, the administration of CoQ10 significantly extended the lifespan, and oral administration significantly increased the CoQ10 concentration in the brains and the mitochondria of test animals. As a result of these promising studies on mice, researchers have been testing the benefits of CoQ10 on humans with ALS. One phase 2 study didn't find any substantial benefit from CoQ10 supplementation in the patients with ALS. However, more research still needs to be done, as CoQ10 plays an important role in the mitochondrial function and controlling the oxidative stress, which are two key components of ALS. In addition, it's been noted that high doses of CoQ10 are generally safe and they may be needed before benefits are seen. Acetyl L-Carnitine has been shown to improve the mitochondrial function. Acetyl L-Carnitine appears to increase the growth and to repair neurons, while also protecting the neurons from high levels of glutamate when combined with lipoic acid. Acetyl L-Carnitine also protects neuron cell cultures from excitotoxicity, one of the putative mechanisms of disease in ALS. Acetyl L-Carnitine has also been found to reduce neuromuscular degeneration and increase lifespan in animal models of ALS. In one animal study, the effects of acetyl L-Carnitine were increased when they were administered in conjunction with lipoic acid. Lipoic acid has been shown to have antioxidant properties as well as to increase intracellular levels of glutathione. It also chelates metals both in the test tube and in animal models. As a result, lipoic acid supplementation might protect the neurons from some of the changes that lead to ALS. Furthermore, lipoic acid has been shown to protect the cells against glutamate-induced excitotoxicity. In one study, the administration of lipoic acid improved the survival in a mouse model of ALS. Adequate protein intake is essential for patients with ALS. Protein supplementation might help to improve the nutrition status of the patients, thereby slowing the progression of the disease. A 2010 study found that patients with ALS taking whey protein supplements had improved nutritional and functional parameters when compared to the control group. Some preliminary data suggest that whey protein might also directly protect motor neurons from oxidative stress, thus delaying the progression of ALS. A Portuguese study suggested that dietary supplementation with amino acids might have some beneficial effects on the course of the disease. In cells, creatine aids in the formation of adenosine triphosphate, or ATP, the primary source of cellular energy. In multiple animal studies, creatine has been shown to provide protection against neurodegenerative diseases. For example, it's been suggested that creatine helps to stabilize the cellular membranes. Creatine may also lessen the burden of the excitotoxin glutamate in the brain, thus improving survival time in animals with ALS. In human ALS patients, there's evidence to suggest that creatine can help to improve the mitochondrial function. In addition, a small preliminary study found that creatine supplementation improved the muscle strength in ALS patients. More recent research has confirmed creatine can protect the neurons from toxic processes such as those that drive the progression of ALS. Creatine, due to its antioxidant and anti-excitotoxic properties, has been found to have a significant therapeutic effect in mouse models of ALS. However, human studies have yielded mixed results, which may be due to insufficient sample size. Creatine can cross the blood-brain barrier and gain access to the brain, a treatment which lowered the levels of glutamate in the cerebrospinal fluid, and this can help to protect the brain. Glutathione is an antioxidant, and it's naturally synthesized by the body. Increasing glutathione levels can help to prevent free radical damage to the cells. The glutathione precursor, N-Acetyl Cysteine, boosts the blood levels of glutathione. Patients with ALS tend to have higher levels of oxidized glutathione, or glutathione that's already been used to protect the body from free radicals. Increasing the levels of glutathione can also protect the neurons from the degeneration in models of ALS. Interestingly, cell culture models have shown ALS is associated with reduced glutathione levels due to mitochondrial dysfunction, and that reduced glutathione levels can result in elevated levels of the toxic glutamate. Along with being a glutathione precursor, N-Acetyl Cysteine has antioxidant activity on its own. In animal models of ALS, N-Acetyl Cysteine administration was shown to decrease the motor neuron loss, improve the muscle mass, and increase survival time and motor performance. In addition, N-Acetyl Cysteine supplementation can help to thin the mucus secretion in the oral cavity, and this makes swallowing easier. Green tea contains high concentrations of catechins, flavonoids with strong antioxidant properties. Green tea extract has been demonstrated to have anti-inflammatory properties. One of the catechins known as epigallocatechin gallate, or EGCG, is of particular interest in the context of ALS. The EGCG and the other catechins might be able to protect the neurons from a variety of diseases. EGCG has been found to protect the cultures of motor neurons from death due to excessive levels of glutamate. Motor neurons can also be protected from mitochondrial dysfunction with the addition of EGCG in culture. The EGCG can also bind to and inactivate iron, and this can help to protect motor neurons from the effects of ALS. Epidemiological data further supports the following role of the green tea for its potential in protection of the neurons. The green tea consumption reduces the risk of neurodegenerative diseases, and people who drink the green tea have a lower risk of developing ALS. Pycnogenol is an extract of marine pine bark, and it includes procyanidins and phenolic acids. It's been shown to have antioxidant properties, as well as protective effects against glutamate excitotoxicity. Pycnogenol is a common complementary therapy for ALS patients. In addition, the pycnogenol increases the levels of SOD. Resveratrol is a powerful antioxidant, and is found in grape skins and Japanese knotweed. Resveratrol has been shown to suppress the influx of excitatory ions into some cell types, which are associated with reduced glutamate-induced cell toxicity. Other ways the resveratrol can target neurodegenerative diseases is by reducing the oxidative stress, both on its own and by increasing the expression of SIRT1, a stress response gene that's associated with longevity and protection against a number of cellular assaults. Although it's not known what role that this gene plays in ALS, increasing SIRT1 expression via resveratrol helps to protect the motor neurons from ALS in a cell culture. In addition, resveratrol can increase the activity of SOD in cells and protect them from apoptosis and oxidative stress. One of the intriguing aspects of resveratrol is it can protect the motor neuron cell cultures from death, which is something that riluzole, the only FDA-approved drug for ALS, cannot do. For more on herbs, supplements, and natural treatment plans, check out my website.